Hi, my name is Carlin Scott, and you're watching Cell Bio News. With SNPs associated with cerebrospinal fluid, phospho-tau levels influence rate of decline in Alzheimer's disease, which is found in the September issue of PLOS Genetics, in a study done by University of Washington, led by Dr. Carlos Cruchaga and Dr. J. Cao. Alzheimer's disease is a degenerative disease of the brain that affects memory. The medical definition of the disease is a progressive neurological disease of the brain that leads to the irreversible loss of neurons and dementia. This disease is the most common form of dementia in the United States, affecting 4.5 million Americans. On a cellular level, the disease is caused by neurofibrillary tangles that form the neurons of the brain, causing microtubules to disintegrate and brain cells to die, thereby releasing a protein called tau, which we'll discuss later, into the cerebrospinal fluid, hereby called CSF. Another protein that was found to be characteristic of the disease is amyloid beta. A beta creates the senile plaques that are present in the brains of AD patients, Alzheimer's disease patients, which also inhibits brain function. On a functional level, there is degeneration in the cortical regions, especially the frontal and temporal lobes of the brain, which control functions such as memory, language, and sensory input. The study focused on levels of a protein called tau, which has been a proven indicator of Alzheimer's disease, in the cerebrospinal fluid of the brain, CSF fluid of the brain also. The protein destabilizes the microtubule network, which leads to decreased axonal transport and to the formulation of neurofibrillary tangles and the death of neurons, which is the cause of Alzheimer's disease. Tau is also released in stroke patients and other brain injuries, although this tau differs in that it is phosphorylated, which is very specific to Alzheimer's disease. Researchers have established p-tau 181 as an endophenotype for AD. That is, it's an internal phenotype specific to that disease. By using tau as an endophenotype, researchers are able to determine if a patient is affected with Alzheimer's. The study evaluated SNPs, otherwise known as single nucleotide polymorphism, in the production and metabolism of tau in association with CSF levels of the phosphorylated tau. The effects of these variants on AD susceptibility, onset, and rate of progression was determined to be related in the phosphorylated tau. It was found that an allele that modifies the tau protein was associated with higher p-tau levels in the CSF, which determines the rate at which the disease progresses. Lower levels of the allele meant higher levels of the p-tau, which means an increase in the onset of Alzheimer's disease in patients. With the discovery of the allele that modifies p-tau, doctors are now able to determine the rate at which the disease will take effect on patients. Researchers believe that creating a drug that will inhibit the tau protein will slow the onset of Alzheimer's disease until there is a cure. Thanks for watching.